Hello, you're welcome to this session where we want to discuss about nerve supply of the GIT. Please, if you have not subscribed, make sure you first subscribe. You can like the video, you can share your comments. When you subscribe, you are able to be notified whenever we release other videos. So today we want to look at the nerve supply of the GIT, whereby it is divided into two. The nerve supply is divided into two, whereby we have the intrinsic nerve supply and extrinsic. And we want to start with intrinsic nerve supply. And intrinsic, you can call it enteric nervous system. Intrinsic because it is within the GIT. And this intrinsic nerve supply, it is provided by two plexuses, whereby we have the myenteric plexus and the Meissner's plexus. Or this one, you can call it myenteric, you can call it Aubach plexus or plexus of Aubach. This one you can call it submucosa plexus. Whereby this myenteric plexus, myenteric comes from the word myo, and myo means muscles. So it is within, it is found in between. This myenteric is found between the outer longitudinal and the inner sacral, between outer longitudinal muscle and inner sacral of the smooth muscles. And its major role is that when we think about food, the thought, smell, taste, and presence of food, it triggers this nerve supply within the myenteric, and they are triggered to release neurotransmitters. And the neurotransmitters which are released from this plexus is that the Aubach plexus releases those that stimulate and those that inhibit. This one is inhibit, this one is stimulate. And those which stimulate, because the major role of myenteric plexus, it plays a big role in GIT motility. It is always responsible for GIT. Either it can stimulate GIT motility or movements, or it can inhibit GIT motility. So whenever it is stimulated by thought, by taste, by presence of food, it is stimulated to release neurotransmitters, and the neurotransmitters that stimulate GIT motility under myenteric plexus. Examples we have like acetylcholine, we have serotonin, and the last one is it is acetylcholine, serotonin, and substance P. So these three stimulate GIT movements, whereby they lead to contractions of these smooth muscles, leading to enhancing GIT motility. So these ones stimulate or increase GIT motility whenever they are produced at the nerve endings of the preganglion nerve fibers. That is myenteric. And whenever we want to stimulate, whenever we want to inhibit GIT motility, we see inhibitory neurotransmitters like vasoactive intestinal peptide, that is VIP, being produced, vasoactive intestinal peptide, or we can see production of neurotensin. So neurotensin and VIP are the inhibitory neurotransmitters. These ones, they inhibit GIT movements, or what we call motility. So that is the role of the myenteric, or what we call the Aubach plexus. Then the second is the Meissner plexus, what we call the submucosal plexus. As its name suggests, we find it in the submucosa layer. As we know that we always have 4 layers: the mucosa, submucosa, masticularis, serosa, or adventitia. This one is found within the submucosa. This one is found in the masticularis. And this one also, whenever we want to increase GIT motility, this one, its role is to increase GIT secretion. It increases, is responsible for GIT secretions. Myenteric is for movement. Meissner's is for secretion of juices in the GIT. And the same thing is whenever there is no food, there is no presence of food, we are not thinking about food, we are not smelling food, we see this Meissner's plexus, or what we call submucosa, it releases inhibitory substances like VIP and neurotensin. They use the same, and these neurotensin are the ones that go and inhibit secretions of the GIT. But whenever we think of food, thought, sight, presence of food, smell food, we see production of the other stimulatory substances like acetylcholine, substance P, and serotonin, leading, and these ones lead to, they stimulate, these ones stimulate GIT secretions, whereby gastric juice can be produced, intestinal juice, all of them are triggered by this. And apart from that, this serotonin also is a it causes constriction of blood vessels. So whenever you see serotonin, is responsible for constriction of blood vessels, or what we call vasoconstriction. So apart from being a neurotransmitter, it also causes a vasoconstriction of the blood vessels. So this is what we can discuss about the intrinsic, or what we call the enteric nervous system. And this enteric nervous system, majorly it works, the pathways of the reflex pathways, they are always 2. It is what we call local and vagovagal reflex. But this one, this one is measured by the local reflex, whereby presence of food causes stretching of the GIT. And when the GIT stretches, it stimulates stretch receptors and chemoreceptors, because this food contains chemicals which stimulate chemoreceptors. Then the stretching of the walls causes the stretch receptors to be stimulated, and they send signals to these plexuses, releasing their neurotransmitters. That is what we call local reflex. Then the vagovagal reflex, we are going also to see it under extrinsic. So let us look at the extrinsic, whereby under extrinsic, we are talking about the autonomic nervous system. And we know that from when we studied about the brain, we saw that autonomic nervous system is divided into 2. Parasympathetic nervous system, and sympathetic nervous system. Whereby parasympathetic nervous system is supplied by the craniosacral is supplied by the craniosacral, whereby in the GIT, we are going to see majorly cranial nerve 7, cranial nerve 9, then we shall see 10, which is the vagus. We shall see cranial nerve 10, which is the vagus. Then we shall see craniosacral, we shall see S1 to S3, whereby these two supplies from the mouth. This is supplies cranial nerve 7, you can call it facial nerve. Then cranial nerve 9 is glossopharyngeal nerve. Those supplies the mouth and the upper part of the oesophagus. Then the vagus nerve supplies from oesophagus up to the upper part of the large intestine. So vagus supplies from oesophagus to the upper part of the large intestine. Then the lower part is supplied by the sacral nerves, S1 up to S3. They supply the lower part of the large intestine, whereby they break from the pelvic nerve. These ones are part of the pelvic nerve, which supplies the lower part of large intestine. And these craniosacral nerves, which are part of the parasympathetic, they are responsible for stimulating GIT motility and secretion. We see the impulses sent by the parasympathetic nervous system that whenever there is need to increase GIT motility, maybe we need a peristaltic movement of the food bolus, it sends impulse via the sensory to the medulla, the brainstem medulla, and then sends impulses via the motor and stimulates the GIT, the muscularis layer, to cause peristaltic movement. And that is what we call vagovagal reflex. So the parasympathetic supply, its major role is to stimulate GIT motility and secretion. So impulses via the parasympathetic nervous system, that is cranial nerve 7, facial nerve, cranial nerve 9, that is glossopharyngeal, cranial nerve 10, that is vagus, and sacral nerve S1 up to S3. They transmit impulses which are responsible for enhancing GIT motility and enhancing GIT secretion. And the neurotransmitter which they use acetylcholine as their neurotransmitter, both at the ganglion and at the muscular junction. They use acetylcholine, which is responsible for stimulating GIT motility and secretions. And lastly is the sympathetic part of the autonomic. And this sympathetic nervous system, for it is supplied via the thoracolumbar. That is from T5 up to L2, is the one that supplies the messages by the sympathetic. And the sympathetic nervous system actually, when you are stressed up, when you have anxiety, that's why we see people who are always stressful. They suffer from PUD because this anxiety causes it stimulates sympathetic nervous system, leading to lack of GIT secretions, leading to no GIT motility, and it can result into PUD. So stress is very dangerous. So we are saying it is supplied by T5, that is thoracolumbar, from T5 up to LT, and the neurotransmitter they use is norepinephrine. They use norepinephrine as a neurotransmitter. We have seen parasympathetic using acetylcholine as a neurotransmitter, but the sympathetic uses norepinephrine, which we abbreviate as NE. norepinephrine. And this norepinephrine, whenever we are stressed out, whenever we are not thinking of food, whenever we are not smelling food, it sends the sensory receptors, sends impulse to the medulla, the brainstem in the medulla, and sends the impulses via the motor neuron, which this motor neuron from the sympathetic nervous system produces this neurotransmitter norepinephrine. And this norepinephrine inhibits GIT motility, it inhibits GIT secretions, and at the same time, it causes constriction of the sphincter muscles. So that is the role of the vagovagal reflex of the sympathetic nervous system. I told you that the reflexes are two, local reflex and vagovagal. Vagovagal meaning the sensory receptor takes the information to the brain, then the motor brings it back. And all of them, we call them vagovagal because they are supplied by the vagus nerve, which is cranial nerve 10. So this is the extrinsic nerve supply of the GIT. So thank you so much. We have seen that the nerve supply in the GIT is divided into two. That is the intrinsic, which is enteric nervous, and extrinsic, which is the one of the brain, what we call the autonomic nervous system. We have seen the intrinsic being done by the myenteric or the Auerbach plexus, found in between the outer longitudinal and the inner circular of the muscularis layer. And we have seen mesenus, which is found in the submucosa. And we have seen the neurotransmitters which stimulate is acetylcholine, serotonin, and substance P. Myenteric doing the major role of GIT motility, mesenus or what we call submucosa GIT secretion, whereby stimulation is by acetylcholine, serotonin, and substance P, then inhibition is by vasoactive intestinal peptide with the neurotensin. Then we have seen in extrinsic, we have the autonomic nervous system supplied by the parasympathetic and the sympathetic. Parasympathetic, that is craniosacral, whereas sympathetic is thoracolumbar. And the impulses transmitted by the parasympathetic, they are responsible for stimulating GIT, motility, and secretion using neurotransmitter acetylcholine. Then the sympathetic uses norepinephrine as a neurotransmitter, and it is the one that inhibits GIT motility and GIT secretion, and also causes constriction of the sphincter muscles that regulate movement of these food substances within the GIT. Thank you so much. Always like, share the comments, and share to others so that we can keep growing. Thank you so much.